Good evening. So in the year 2002, after the Moi era, the Kibaki government came up with a Vision 2030 in which they intended to transform the Kenyan economy from agricultural-based economy to a fully industrialized economy. So tonight, I want to talk about industrialization because we are only 5 years away from 2030 and I don't see any of that happening. So I'll talk specifically about pharmaceutical companies or pharmaceutical industries here in the United States and see how medicines are discovered, invented, and manufactured. To understand a drug and how they work, you have to understand a disease. I'll not spend a lot of time talking about diseases, but in general, interference of normal body functions by microorganisms, chemicals, or genetics results in a disease state. When microorganisms gain access into the body, they compete with the body cells for nutrients. Some microbes feed on the body cells, while others modify the functions of the body. The metabolic byproducts of some of the microbes inside the body may interfere with the normal biological process. For example, a botulinum toxin produced by a Clostridium botulinum, which is a potent neurotoxin. The body cells have surface transmembrane proteins which perform functions like signal transduction, enabling intra and intercellular communication. Transmembrane proteins and signaling molecules, that is the hormones, play an important role in modulating biological activities within an organism. Biological functions are usually fine-tuned, and any slight interference leads to a disease. A signal molecule, e.g. insulin, is released by the pancreas, a member of the endocrine system, and it interacts with the insulin receptors, hence signaling the cells to start the uptake of glucose. The result is reduction in glucose level in circulating blood. Diabetes results from the inability of the body to regulate blood glucose level. This can be due to insufficiency or no insulin from the pancreas, that is type 1 diabetes, or it can be insulin resistance, where the receptors do not bind to the insulin molecule, that is the type 2 diabetes. So understanding how insulin interacts with the insulin receptors has led to development of insulin in the laboratory, hence opening a multi-billion-dollar market. So how do they do it? They start by understanding the physiological process at the cellular and molecular level. They then identify the druggable targets, in this case either the insulin receptor substrate or the insulin itself. They understand the molecular interaction within the druggable targets by determining the biochemical properties as well as identifying active binding sites of the target. Those who have a background in biochemistry know that proteins folds in a specific way, and the main secondary protein folding structures are the alpha helices and beta pleated sheets. They form a tertiary structure which, depending on the proximity of specific amino acid, can have affinity for molecules with the specific surface attributes. There are equipment that can be used to measure the binding affinities, for example, the SPR, that is the surface plasmon resonance, in which a gold plate is coated with molecules of interest, and when the molecule that is being tested flow over the molecule of interest on the gold plate, if there is binding, a current is generated which is converted into a series of values that can indicate the binding affinity constant, that is the KD. These molecules, which are now drug candidates, are then tested in the laboratory animals with the disease model to evaluate their safety and efficacy. The research scientists investigate if the drug is able to suppress or cure the disease. This is called a preclinical trial. And when the data shows that the drug is safe A pharmaceutical company can then apply for IND, that is Investigational New Drug, to the FDA for license to conduct a phase 1 clinical trial. So in a phase 1 clinical trial, the pharmaceutical company recruits about 20 to 50 or more healthy people, depending on the availability of people that are willing to participate. Half are given the drug, while the other half are given a placebo in order to evaluate the safety of the drug. Statistical analysis is performed to determine if there is a difference between those who took the drug and those who took the placebo. If there is a difference, the p-value is calculated to determine whether the difference was statistically significant or it occurred by chance. When the phase 1 clinical trial is completed and the pharmaceutical company submits the data to the FDA reviews the data and determine if the company can proceed with the phase 2 clinical trial. So during the phase 2 clinical trial, the pharmaceutical company works with the doctors and hospitals to recruit real patients, normally over 100, and they are divided into 2 groups, just like in phase 1 clinical trial, and half of the patients are given the drug while the other half are given the placebo. So the data about efficacy and safety is collected. Statistical analysis is performed between the 2 groups of patients to see if there was a improvement between the group that took the drug versus those who took the placebo. Theoretically, those who took the placebo should not see any improvement, but some normally most of those who took the drug should see improvement if the drug is efficacious. Since the patient participating in phase 2 clinical trial do not know whether they took the placebo or the real drug, some of the patients on placebo might come up with side effects, since some people are opportunistic and can demand to be paid by the pharmaceutical company for having an intended side effect. If the drug shows that the difference in the improvement between the 2 groups is statistically significant, they submit the data to the FDA and apply for phase 3 clinical trial. This now covers a wide range of patients, including young, old, as well as many races. When the data shows that the drug leads to improvement in disease condition, the pharmaceutical company now compiles and submits the data to the FDA with the drug instruction insert, that is labeling for application to market the drug. This is now called the application new drug if it's a small molecule, or biologics license application for biologics, e.g. the vaccines. Before the pharmaceutical company can manufacture and market the new drug, they have to develop the manufacturing process and have that process validated by the FDA. Then the manufacturer will go ahead and manufacture the master batch and keep all the records to show that all the critical quality attributes of the drug are within acceptable range and there is no statistically significant variance within the batches. The pharmaceutical company then submits all the data to the FDA, and when all is in order, the FDA grants approval to manufacture and sell the drug in the United States. The test now enters phase 4 clinical trial, where the data is collected for a very large population, and if there is a statistically significant effect on the drug, this is called continuous monitoring. The whole process from discovery to approval by the FDA can take up to 15 years and billions of dollars. So the FDA can grant exclusive rights to manufacture and sell the drug for up to 20 years. after which, if the exclusive right, that is the chemical patent, is not renewed within the time frame, other companies can come, obtain the formula and manufacture the so-called generic drug, basically a copy of the brand name. Therefore, whenever you take a drug and it works, there are many hours of intense labor and frustrations to get that drug to our pharmacy.